Hello students, today we will be dealing with a very basic topic called as the structure of enzyme. I am Syed from Department of Studies in KSOU. The men who matter. Actually, these are the scientists who first proposed the structure of an enzyme. Sir Emily Fisher was the one who gave us the lock and key hypothesis, and Sir Daniel E. Koshland is the one who gave us the induced fit model, which is even accepted till date. Enzymes Enzymes are the substance that acts as a catalyst in living organism. They regulate the rate at which chemical reactions proceed without itself being altered in the process. So when a enzyme catalyzes a reaction, it does not alter itself. Here we are seeing a 3D structure of a protein that is an enzyme. Enzyme is a protein, and we are seeing the 3D structure of an enzyme. Enzyme has two active sites. One is where the substrate can bind in. The blue one shown in the image is a substrate binding site, and the red one is an catalytic site where the inhibitors can come and bind. Now, when an inhibitor binds here, it reduces the chemical activation energy. Okay, and thus no product formation can happen. So this is the basic structure of an enzyme. Currently, the 3D model having the two active sites. One is the binding site, and another one is the catalytic site. Now let's study the structure of the enzyme in little detail. So what are enzymes made up of? A large protein enzyme molecule is mainly composed of one or more amino acids chain called polypeptide chain. The amino acid sequence determines the characteristic folding pattern of the protein structure, which is essential to the enzyme specificity. This amino acid sequence actually determines the characteristics of the enzyme. The active sites gets its property from the amino acids it's built of. So whatever amino acids are present in the active site, they help in the characterizing the active site, the which type of substrate it will attract, or which kind of substrate it has affinity to. This is decided by the amino acid sequence present in the active site. These amino acids may have side chains that are large or small, and these side chains can be either acidic or basic, hydrophilic or hydrophobic in nature. If the enzyme is subjected to changes such as fluctuation in temperature or the pH, the protein structure may lose its integrity. That means it can denature, and this enzymatic ability of this enzyme will be altered. Denaturation is sometimes is always irreversible. The set of amino acids found in the active sites, along with their position in 3D structure, gives the active site a very specific size, shape, and chemical behavior. Thanks to these amino acids, an enzyme active site is uniquely suited to bind to a particular target, that is the enzyme substrate, and helps them undergo a chemical reaction. Therefore, we can tell that the amino acids which are found in the active sites are the one which makes the enzyme highly specific to its substrate. Now we are looking at the lock and key model given by our scientist Emil Fisher. So what is lock and key mechanism? Lock and key mechanism was first proposed by Emil Fisher to explain the binding between the active site of an enzyme and the substrate molecule. The active site was thought to be a fixed structure, just like a lock, how it is rigid. Same way, the active site was thought to be very rigid with exactly matched structure of the specific substrate the key. Here, both the structures of the substrate as well as the active site of an enzyme is considered to be very rigid in the nature. To proceed further, the lock and key hypothesis is mainly focused on the active site. The active site of an enzyme has a very unique geometrical shape and it is complementary to a specific substrate molecule. Imagine a puzzle piece. There are only few pieces that fit with one piece because the active sites are so geometrically unique that the enzyme can work with only few or just one substrate. In order to explain why enzymes have such a high rate of specificity, Emil Fischer suggested that both a substrate and enzyme have specific geometrical shape that fit exactly into each other. This idea of both substrate and enzymes having a natural geometrical fit is called as lock and key hypothesis of Emil Fischer. Here we can see a diagrammatic representation of a lock and key model where different substrate come and fit into the enzyme. Both the enzyme and the substrate have not changed any of their features. Their shape is rigid. The enzyme is unaltered and the product is formed. Okay, thus the lock and key hypothesis of Emil Fischer. This work brought Emil Fischer fame. The Nobel Prize in Chemistry in 1902 was awarded to Hermann Emil Fischer in recognition of extraordinary services he has rendered by his work on sugar and purine synthesis. Okay, now let's see the another hypothesis given. So in contemporary times, there is another hypothesis given by Daniel E. Koshland, and this hypothesis is called as Koshland's induced with hypothesis. This hypothesis is an offshoot. That is, it is different from the previous model. The current model supports a more refined view of enzyme substrate binding. As the enzyme and substrate come together, their interaction causes mild shift in the enzyme structure that confirms an ideal binding arrangement between the enzyme and the substrate. This dynamic binding maximizes the enzyme's ability to catalyze the reaction. Okay, now you can see in this image that this is an enzyme and it is its active site. When the substrate comes and binds to the enzyme, there is a clamp down effect in the enzyme confirming an induced fit model. It is just like the enzyme is hugging the substrate. Okay, induced fit. Okay, you can see here how tightly it is binding. It is because of the clamp down effect of the enzyme, where the enzyme is clamping down its substrate. Okay, so here is an image where there is an enzyme and its active sites. When the substrate comes in contact with the enzyme slightly changes its active site conformation so that it can bind the substrate properly. The enzyme substrate complex is formed, then the enzyme product complex is formed, and then the products are released from the enzyme, forming the and the enzyme is again regaining its original active site. Okay, so now let's go in detail. The matching between an enzyme site and the substrate isn't just like two puzzles fitting together, though as it was said in the old model of lock and key. Instead, an enzyme changes its shape slightly when its substrate comes in, binds in, resulting in even a tighter fit. This adjustment of the enzyme to fit the substrate is called as an induced fit. When an enzyme binds to its substrate, we know that it lowers the activation energy of the reaction, allowing it to happen more quickly. But you may wonder, what does the enzyme actually do to the substrate to make the activation energy even lower? The answer depends on the enzyme. Some enzymes speed up the chemical reactions by bringing the two substrates together in the right orientation. Others create an environment in the active site that favors to the reaction. The enzyme substrate complex can also lower the activation energy by binding the substrate molecules in such a way that it facilitates bond breaking, helping to reach the transition state. Finally, some enzymes lower activation energy by taking part in the chemical reaction themselves. This is the active site. Reduces may form temporary covalent bonds with the substrate molecules as part of the reaction process. But this is just temporary. In all cases, the enzyme will return back to its original state at the end of the reaction. It won't stay bonded to the reacting molecule. In fact, a hallmark property of the enzyme is that aren't altered by the reaction they catalyze. When an enzyme is done catalyzing a reaction, it releases the energy that through the products and is ready for the next cycle of the catalysis. Thus, the induced fit hypothesis of the Koshland is explained. Now, students, let's look the differences between the induced fit model and the lock and key model. In the induced fit, the binding is flexible. The active site is no longer rigid structure. Here, the binding is very strong. The rigid structure of the active site is a hallmark of this model. But in induced fit, the active site of an enzyme is dynamic, undergoes a conformation change to improve binding. Here in lock and key model, the active site of an enzyme is static. No conformational change can happen at the site. The induced fit model explains the mechanism of enzyme structure binding when they are not complementary in the shape with each other. This lock and key model explains the binding of perfectly matching substrate and enzyme with each other for the reaction. Induced fit model shapes are not complementary with each other before binding. In lock and key model, the shapes are complementary with each other before binding. Thus, are two models, induced fit hypothesis and lock and key hypothesis of enzyme structure. Thank you.